Hello everyone and good morning. This is Dr. Sudarshan Samajura Pedge, Assistant Professor, Department of Zoology, Kailas Rasika Mahavidyalaya Devne, your zoology companion, and welcome to my lecture. In the last lecture, we also completed the introductory part of carbohydrate metabolism as well as the one important process or one important cycle we also studied, that is called the glycolysis. After that, today we will focus on another topic of our syllabus, that is glycogenesis, glycogenolysis, and glyconeogenesis. Okay, so let's get started today's lecture. First is the glycogenesis. In the glycogenesis, glyco means glycogen and genesis means formation. Okay, in that process, that is the one particular organ in our body, it will be the genesis, that is called as the liver, is a one particular organ that all cells of animal body are capable of storing glycogen, but mammalian liver and muscles can store glycogen up to 15 to 20% of their weight, okay, as compared to the other cells. Okay, it means the glycogen storing organ is the liver as well as muscle cells. Okay, and that liver glycogenesis is an anabolic polymerization of glucose into glycogen. Okay, you have seen here the glucose is first phosphorylated in the presence of enzyme glucokinase, right, and magnesium ion, okay, forming glucose 6 phosphate. Okay, right. After that, then glucose 1 phosphate by phosphoglucomutase. Okay, you have seen here phosphoglucomutase enzyme form glucose 1 phosphate, right, and next into the uridine diphosphate glucose, right, with the help of phosphorylase enzyme, right, and then finally enzyme glycogen synthetase changes it into glycogen. Okay, it means the glucogen is converted into the glycogen, and it means the large amount of glucose we have taken in at the time of feeding, that time some glucose, it will be stored in the form of glycogen, and where it stored, that is the one particular place. That place is called the liver as well as the muscle cells. Okay, it means the glucose is conversion into the glycogen and storing in the liver as well as muscle cell. Okay, that total process is called as glycogenesis. Okay, next process is the glycogenolysis. In that process, the process of breakdown of glycogen is called glycogenolysis. Okay, glyco means glycogen and lysis means the to break down. Okay, it means the glycogen, it is completely break down into the glucose, that is called the glycogenolysis process. Okay, when the blood sugar level falls, when it will be occur in the glycogenolysis, at the time of blood sugar level falls, glycogen stored in the tissue, especially glycogen of kidney, okay, liver and muscles may be breakdown. Okay, at the time of blood sugar level, it will be the falls down. Okay, that time glycogen stored in the tissue as well as in the kidney and liver and muscles also, that similar time it will be breakdown. Okay, that is the one particular process, that process is called as glycogenesis. At that time, glycogen may be broken down to glucose in liver and kidney, or it may be broken down to glucose 6 phosphate in the muscles. Okay, so there are three types we have seen here, or there are three steps we have seen in the we have see here the glycogenolysis process. That the first step in the breakdown of glycogen is catalyzed by two enzymes you have seen here, which act independently, right? The first enzyme, namely glycogen phosphorylase, inorganic phosphate, catalyzes the cleavage of terminal alpha 1,4 bond of glycogen to produce glycogen with glucose molecule. Okay, you have seen next slide. That is the one particular glycogen phosphorylase. It will be the cleavage of terminal alpha 1,4 bond of glycogen to produce glycogen with one glucose molecule less than, okay, that is the n minus 1, right? Glycogen n minus 1, it is the one glucose molecule is less. Okay, the removal of glucose residue as a glucose 1 phosphate. Okay, the removal of glucose residue as a glucose 1 phosphate continues until about 4 glucose residues remain. Okay, so that is the one particular glucose 1 phosphate, it will be the form. Okay, another next second step. In that second step, you have seen here the glucose 1 phosphate is then reversibly converted to glucose. Okay, you have seen here the glucose 1 phosphate is then reversibly converted to glucose 6 phosphate by the action of enzyme phosphoglucomutase. Okay, you have seen here that reaction is the reversible reaction. Okay, and it will be the form in the glucose 6 phosphate, right? That is the one particular second step of this glycogenolysis. Okay, another third step, the next reaction you have seen here, namely the conversion of 6 phosphate to glucose, right? 6 phosphate to the glucose, right? Takes place in the liver and kidney by the action of enzyme glucose 6 phosphatase, right? This enzyme removes phosphate from glucose 6 phosphate and enabling the free glucose to diffuse from the cells. Okay, that is the one particular formation of glucose. And such type of process, it will be formed with the help at the type of sugar levels is falls down. That time glycogen is converted into the glucose. Okay, third process, that is the gluconeogenesis. Question may arise, what is meant by the gluconeogenesis? Okay, the synthesis of glucose from non-carbohydrate precursor is known as gluconeogenesis. Okay, it means at the time of formation of glucose with the help of non-carbohydrate precursor, and that total process is called as gluconeogenesis. Okay, after the mechanism involved in the gluconeogenesis is the reversal of citric acid cycle and glycolysis. We know very well the glycolysis process. In the glycolysis process, we have seen that the glucose is converted to the pyruvic acid. Okay, that is the one. That process is called as glycolysis. But in that, the pyruvic acid is converted into the glucose. That is a reversible process of glycolysis. That process is called as gluconeogenesis. Okay, so where does gluconeogenesis occur? The major site of gluconeogenesis is liver, okay, and kidney also, okay. Why? Because it is only about 1/10 of that form in the liver because of its smaller mass, okay. So the brain and skeletal muscles have a high demand for glucose. Very little gluconeogenesis takes place in the brain, okay, and skeletal muscles and heart muscles also, okay. Gluconeogenesis by liver and kidney serves to maintain the glucose level in the blood, okay, so that the brain and muscles can extract sufficient glucose to their metabolic demand, okay. It will be the occur generally in the liver as well as kidney, okay. When does gluconeogenesis occur? At the time of carbohydrate in the diet is insufficient carbohydrate in the diet, that time gluconeogenesis it will be occur, okay. The intake of protein, second is the intake of protein rich diet resulting in the absorption of large amount of amino acid. For example, animals such as carnivorous animals which completely rely or which completely depend on high protein diet, okay. So that time gluconeogenesis it will be the occur, okay. Another next the at the time of starvation, okay, when the tissue proteins are broken down to amino acid, that time gluconeogenesis it will be the occur, okay. And last is the fourth in the during heavy muscular work, the large amount of lactic acid is produced by glycolysis, that time gluconeogenesis it will be the occur, okay. That is the one particular four categories at the time gluconeogenesis process it will be the occur. First is the at the time of insufficient amount of or even insufficient carbohydrate in the diet, that time gluconeogenesis it will be occur. Other next is the intake of protein rich diet, right, large amount of amino acids, that time gluconeogenesis start or occur, okay. Next third is the at the time of starvation upasmar, that time is the gluconeogenesis it will be the occurred. And number fourth is the heavy muscular work it will be the occur, that time gluconeogenesis it will be the starts, okay. It means the synthesis of glucose from non-carbohydrate precursor is known as gluconeogenesis, okay. That is complete process is called the gluconeogenesis, okay. So